Hello, everyone. Good afternoon. Here in Arizona is the afternoon of Monday, June the 15th of 2026. Today, I wish to share with you a new study published by Japanese scientists that sheds a new light on the relationship between the microbiome and behavior. This is the study. It is entitled Gut Microbiome Composition and Predicted Functions Relate to Growth and Behavior in a Japanese Preschool Cohort. And the subtitle for today's short presentation is that in preschool age, that is a critical window in which the microbiome stabilizes and the brain matures rapidly, even small variations in behavior are associated with distinct changes in the microbiome. Well, let me tell you that we have been working on the relationship between the microbiome and autism since 2014, when I first went to the Autism One conference in Chicago. I couldn't find the actual slide of 2014, so I found this one of 2015. And we have attended the Autism One and other autism conferences for many, many years. So we have studied the role of the microbiome in autism. And with this slide, I just wish to summarize what is known now in 2026. It is known that there are alterations of the gut microbiome in the spectrum. They have been documented for over a decade. The results, of course, are heterogeneous because of differences in age, diet, geography, GI comorbidities, and the methodologies of analysis. But nevertheless, some recurrent patterns emerge from the most recent meta-analysis, which means the study of many studies and reviews. In children with autism, there is a reduction in the biodiversity of the microbiome. A depletion of beneficial bacteria, such as the bifidobacterium, that here is highlighted as a mistake. Forgive me, but I don't know how to use this new platform very much. So it's a kind of amateurish as far as this slide presentation is concerned. But I think that what is important is the message that there is also depletion of the bacteria producing short-chain fatty acids, in particular butyrate, butyric acid. There are alterations in the metabolism of bile acid and glycans, and there are signs of increased inflammation or immune dysregulations, and all these signs are frequently observed in autism. As a matter of fact, if you transplant the microbiota from donors with autism into mice that are germ-free, so they can receive such a transplant, you can induce the behavioral phenotypes of autism because of the microbiota, as if the culprit for autism were the microbiota. And this provides a suggestive indication that the microbiota alterations are the cause of autism. However, the things are not so clear-cut because there is a debate still going on how much these alterations are a primary cause of the clinical picture or they are a consequence of the dietary habits typical of autism that are selective and repetitive, or a mix of both factors. But nevertheless, we know that if you transplant the autistic microbiota into germ-free, let's say healthy from the point of view of behavior mice, they become autistic, so to speak. So definitely there is a role. And here comes this Japanese study that was published just a few weeks ago. This is the reason why I wanted to bring this to your attention, because it's a fundamental contribution to the typical sample. So these alterations of the microbiome and their role in shaping the behavior are not limited to autistic children. They happen in everybody, including ourselves, and they are more pronounced during childhood. So this study was conducted, as I said, a few weeks ago. Well, it was published a few weeks ago, and it was conducted on 36 non-autistic, normal, neurotypical, as we say, Japanese preschool children without any type of pathology. Their mean age was 48 months, and they were recruited from low-risk communities in Japan. And this study fits with particular relevance because what did the researchers do? They analyzed the gut microbiome through genetic analysis, the so-called 16S rRNA, sequencing those genes, and they predicted the metabolic functions. They measured the growth parameters of these children and evaluated the behavior with a questionnaire that is a standardized one. And this questionnaire explores internalizing, externalizing domains, attention, withdrawalness, somatic complaints, and sleep. So the same parameters, more or less, that are used for the diagnosis and the evaluation of treatment of autism. But again, these were not autistic children. These were neurotypical children. Now, unlike most of the literature on autism that focuses on a clinical or high-risk samples, this work examines mild behavioral variations because, of course, we are not all the same, and the behavior of these children varied, was different from child to child, but still without any diagnosis of neurodevelopmental disorder. And precisely for this reason, this study represents a precious baseline. It allows an understanding of which microbial mechanisms are already active during a typical neurobehavioral development and how this same mechanism might be amplified, distorted, or combined with other factors in autism. So this study demonstrates that the involvement of the microbiome in behavior is not a phenomenon limited to pathological pictures, but it is an integral part of early neurobehavioral development. Now, during the preschool age, that is a critical window in which the microbiome stabilizes and the brain matures rapidly, even small variations in behavior are associated and were measured with distinct microbial configurations, which means with distinct alterations of the balance of the microbiome. And these results reinforce the model of the microbiota-gut-brain axis that is a continuous and dimensional phenomenon. It's not a dichotomous one, as we say in science, that is either healthy or pathological. No, it's a continuum of changes, and the changes in the microbiome are associated with variations in the behavior. Because the study shows that patterns partially overlapping with those described in autism already exist as correlates of mild variations in typical children. For example, the reduction in bifidobacteria that corresponds to a reduction in the fermentative capacity in children, in neurotypical children, was associated with higher withdrawalness scores. And this is similar, recalls the depletion of bifidobacteria and butyrate producers that is observed in autism, where in autism, such a depletion is associated with social difficulties and GI symptoms. So essentially, in these neurotypical children, the Japanese scientists, they observed that alterations of the microbiome were associated with mild changes in behavior, but the same type that are exaggerated in autistic children. And in synthesis, the Japanese study convincingly reinforces the model of the microbiota-gut-brain axis in the early development of our brains, of our behavior. Demonstrates that microbial mechanisms that are partially overlapping with those of autism are already operative as correlates of mild behavioral variations in typical children. And therefore, autism can be interpreted, at least in part, as an extreme expression or a multifactorial perturbation of these same axes, amplified by genetics, early environment, diet, the diet of the mother during pregnancy, and other factors. The study reinforces the scientific rationale for targeted interventions on the microbiome. Things that we know, that we are aware of, a diet rich in fermentable fibers, probiotic strains such as those that are depleted, such as bifidobacterium, prebiotics that help the balance of the microbiome, both in clinical contexts such as autism, but also potentially in all children, and in particular in children with subclinical traits of internalizing, withdrawingness, or disturbed sleep. And this Japanese study, in my humble opinion, remains a fundamental observation precisely because it reminds us that dialogue between gut and brain begins long before the appearance of overt clinical pictures. I thank you so much for your attention. I hope that you have enjoyed this short presentation of mine, and I am determined to present new studies on the microbiome and other topics as well on a regular basis. Thank you so much.